Hello, everyone. We're here with Professor Harris Wang, who is an assistant professor in the Department of Systems Biology at Columbia. And we are the Synthetic Biology Initiative at Columbia University. And I'm joined here by my colleagues, Benji Greenfield and Brandon Cuevas. And we're going to ask Professor Wang a couple of questions, and we're going to see what he thinks about systems biology and synthetic biology.
Who are you and what are you working on?
My name is Harris Wang. I'm assistant professor in the systems biology department, and also have a joint appointment in pathology and cell biology at the medical school. And we're working in the area of synthetic biology, and so we specifically are interested in designing and building microbes that could colonize a variety of environments, and to be able to understand how those environmental conditions change and how that can affect those microbes, and use those microbes to perform interesting new functions. Some examples that we're pursuing, for example, are things associated with the gut microbiome, where you could potentially think about deploying kind of these engineered probiotics. You might be interested in using some of these things also in soil or in other environments that are interesting. So we mostly focus on microbes, although we do have some kind of projects also in engineering eukaryotic systems as well.
So thank you for your input, Dr. Wang. So our next question is, what is your conception of what synthetic biology is? So in other words, how would you define this rapidly evolving field?
I think that's a great question. I get that question a lot, actually, from people in and outside of the field. And there's actually many different definitions that people adopt. And so maybe I'll give you a couple of them, and I'll give you my take on that. And so the first is, when you think about synthetic biology, you think about maybe artificial life. So things like having a cellular system that just has completely non-natural properties. For example, they have, instead of having just A, T, C, and T for their genetic code, their DNA, they might have expanded genetic codes, or they might have ribosomes that could produce new proteins by incorporating non-natural amino acids. And so those are things where they're almost like alien life, right? And so that's one area in the field thinks about synthetic biology in that context. Another kind of definition is really one where you're thinking about engineering biological systems, but in a very kind of defined and sophisticated way. And I would say really it's very similar to how we think about kind of the semiconductor industry, where you're not just kind of tweaking with something and then playing around with it and trying to get a result, but rather you're formulating a particular design a priori, having the knowledge to be able to incorporate that into a design modeling framework, and be able to test and manufacture those designs, and to be able to generate kind of new things out of that. And so you're thinking about an iPhone or whatever, right? So that that gadget really requires a lot of a priori design and planning and manufacturing to produce. And we really want to do the analogy of that in biology. And so another way that people have thought about and are working on in synthetic biology is to bring this kind of more rigorous engineering discipline into what traditionally is a biology field, or kind of manipulating biological systems for useful purposes. And so in that context, it's really about kind of engineering biology in a kind of very structured and very disciplined way, where we can actually be able to do that very well, as opposed to just kind of doing a lot of tinkering without having this higher level of abstraction, which I think is really needed if you're thinking about how do you build sophisticated systems. So, one example is that, these layers of sophistication really makes the complex gadgetries that we're accustomed to. For example, the phone, can be decomposed into many hundreds of layers of abstracted architectures, and all of those architectures together, built on one another, makes the overall products very interesting and useful. And biology is built in some ways based on the same level types of architectures. You have genetic information that's encoded into the DNA, which then gets produced to RNA and then proteins, and those proteins interact with one another to build cellular organelle, and as well as the whole cell, which then connect together into tissues and to actual organs and into the whole kind of cellular organism. And so those are the types of things that we like to be able to understand how to abstract down into an engineering discipline. And so for me, I guess synthetic biology is perhaps a combination of those two things. We're certainly very interested in building technologies for engineering biological systems in a way that's more reproducible, more robust, and that's more predictive. But also at the same time, we like to be able to not just produce some new compounds, but also understand the underlying kind of architecture of what constitutes life, and how do you push the boundaries for defining that or redefining it in a synthetic context. So could we really try to engineer cells that could do functions that really aren't manifested in nature? For example, getting cells to be able to record some event that is going on and be able to act upon that event in some very directed way, and be able to evolve based off of that. And so all of those things, I think, are much more kind of where the future of the field lies. And so that's kind of my take on that.
How did you get involved in synthetic biology, and what made you interested in your subfield of synthetic biology?
So, if you think about when the field started, I guess you could kind of go back. It's probably hasn't been around for not much more than a decade. And so actually, I was introduced to the field when I started graduate school, and I was looking for really an area where I can apply kind of my interest and my passion. And I've always had an interest in understanding biological complexity, evolution processes by which simple agents could come together to form very complex properties. And so, that's perhaps a more kind of physical or, way of looking at this. But certainly, when I was in grad school and I had started working in George Church's lab, that's when kind of there was a really strong push in this area by a number of kind of individuals kind of in the early days of the field, where, let's say, could we think about engineering biological systems using this kind of black box approach, and to bring in the abstraction, a layer of kind of architecture to think about engineering biological systems. And so for me, that I felt that was really compelling as a way to think about not just making biological systems or building them, but rather testing and understanding the underlying architectures of what makes biological systems what it is and makes it robust. It's, what are the most complex kind of system that's out there. It can certainly do things that we can barely start to dream about, and so certainly a lot of inspiration are adopted from biological systems. And so for me, that was my route in that. And so the first thing that I really started working on was to work on this project where we tried to recode the genetic code of a bacteria. And so the idea was that if you could change the genetic code, you've essentially produced a very different microbe. So that was a very compelling starting point for me because it had very kind of fundamental roots, but also had very interesting practical applications. Because if you could do that, you could potentially engineer the microbe to do a variety of other, even more practical and useful things. For example, being able to alter its metabolism to be able to produce a variety of kind of bioindustrially useful compounds and therapeutics. And so that's kind of where I started on that, and then kind of haven't left since then. So I think this field has really grown tremendously over the last decade in terms of thinking about how do you design and model these systems and manufacture and test and be able to iterate on that. I think some of the earlier things in the field have been really toy systems, things where let's see if we can build a oscillator that could kind of flip back and forth, or some type of switch, or very very simple toy systems that describe something kind of simplistic, but also something very important and fundamental. And so now we're really transforming that into additional layers of complexity. So making kind of much more sophisticated genetic circuits. So that's kind of where things are, I think.
All right. So the next question is, what is the overarching end or like long-term goal of the field? So what are, in general, what are synthetic biologists working towards, and what are they trying to achieve?
Yeah, so I think the field has a lot of potential, and it really is where I think we're just at the tip of the iceberg here. The field, I think, is really, there's a one part of the field that thinks that, if we build the right tools to be engineer biological systems very easily and rapidly, we could do almost anything, right? Anything that where biological system interfaces with our everyday environment, with our health, with the infrastructure that we build, is going to be important. And so I think in some ways the field is trying to build the underlying technologies to get there, right? And then once you have that technology, you could do whatever you want with it. It's almost like, what do you do with a computer? Well, someone has to build the first computer, and once you build it, I don't think we could have necessarily predicted all of the things that have come from it since then, right? But I think the necessity to have a system by which we can generate that set of kind of engineering disciplines is really kind of where the field is pushing towards. I can name a few what I think are maybe more near-term applications that we'll probably be seeing in the next few years, 5 to 10 years, and I would say those are revolving around using engineered bacterial or mammalian systems for in the healthcare sector in terms of being able to fight diseases, be able to diagnose them, and be able to kind of treat them. And so certainly from a microbial perspective, you could imagine getting bacteria that could sense and report on kind of negative health kind of events. For example, you have inflammation in your gut, perhaps you could get a bacteria that could tell you this and be able to produce anti-inflammatory molecules, right? Certainly in terms of therapies, there's a lot of kind of interest and a lot of activities in things like immune oncology, where you're engineering T cells to be able to recognize and kill cancers. And you do this by, putting in specific kind of markers, and that allows the cells to be able to hone in on the specific kind of tumor types that are people are interested in. And so those are kind of more therapeutic applications and health related. There's a long-standing industry which is the metabolic engineering industry for biomanufacturing for a variety of compounds. Anything that you can think about making it with chemistry, you can potentially think about making through a bug or through a bacteria or yeast or plant cells or even mammalian cells for biologics and drugs. And so thinking about and how do you design and plan and develop strategies to be able to produce a variety of different chemical compounds as opposed to having to naturally source them from kind of difficult environments. For example, extracting artemisinin from a plant to fight malaria may be very difficult, but if you get a bacteria to be able to produce it, that's a much more viable path, right? And there's many such examples. And so but also in other areas, for example, in agriculture, right? Engineering bacteria that could improve the performance of crops and be able to prevent detoxify contaminants that are in the soil or in other environments is an area that's going to be poised to be really disrupted by kind of advances in this field. As well as other things like materials, right? So you could start thinking about using these engineered biological systems to start to build maybe materials that are very difficult to manufacture. Certainly, look at a seashell, and it's made out of kind of very hardy material that lasts for a lifetime and more. For the organism, and how do you start to think about designing them in a programmatic way? And I think in the future we could think about, well, instead of growing a tree, could we grow a house? And how do you get a plant to be able to produce a specific shape of interest, and such that you really have to do very little on the actual production side, but rather put a lot of time and effort into designing it so that it could develop morphologically into a defined shape, which is quite robust if you look at kind of, everything that we're surrounded with. We all reproducibly have 10 fingers and 10 toes, and it's through a very specific genetic program. And if we could somehow be able to engineer those, I think that would be incredible. So.
What do you think is holding us back in synthetic biology?
So if you're asking about kind of what are the challenges that are being faced by people in the field, I think it comes in a couple of flavors, I would say. One, I think, is one about manufacturing, right? We can potentially make designs. We can design things in the lab, but making them is actually kind of challenging, right? So be able to synthesize DNA and be able to put them together into long pieces that then we can boot up in a cell, for example, is still one of the rate limiting steps. And so if we could drastically reduce the cost of DNA synthesis, gene synthesis, and increase the fidelity and the rate we could do this, I think that will really kind of be a watershed moment for the field. What you really want is to have some type of desktop synthesizer where you can kind of type in the genetic sequence of interest and be able to have it spit out the actual sequences for you to test in a cell. And so we're not there yet. Certainly there are advances in the field that will push us into this field, into this kind of vision. I think that's also the distinguishing thing about synthetic systems versus natural systems is that we are ordering the nucleotides in a very defined way that may or may not exist in nature. And so that process is inherently challenging, right? If you look at all of biology, there it's all reproductive in that you're replicating a piece of DNA, and sometimes you make errors, and so then you kind of accumulate those errors, and maybe those errors are functionalized, but you're not really doing this kind of de novo putting nucleotides together in some very defined way. And so that's has from a physics perspective, the thermodynamics of that is very challenging, right? You're essentially drastically changing the entropy of the system, right? Reducing it from something that's completely random to something that's it's very ordered. And so there's some inherent challenges in this, which I think are technical challenges that I think, as well as scaling challenges. So more people start to order more DNA, it will become cheaper, right? Just it's an economic argument there. So that's one, I think, major limitation the field faces. I think the second is one of maybe more related to just the underlying complexity of biological systems, right? That you can only design what you know, right? And the processes by which those things are obeyed in natural systems. And so there are a lot of things about biological systems that we just don't know yet. That includes not just all the different components within the cell, even the standard and very the best studied bacteria, E. coli, still has 25% of its genes that are not known what their functions are, and so there's a knowledge gap in all of this. But also, I think there's just a conceptual gap of how do we design complex, messy systems that are working asynchronously, and what are the languages to describe and to predict their behaviors. And so that's a much bigger topic about like emergent properties themselves. But I think we've already come very far in this area, and I think with new technologies such as things like genetic engineering, CRISPR, and these other systems that really kind of has opened up kind of fundamentally new areas for us to test. So I would say even though the limitations are present, I think we're going at a much faster rate in advancement than probably even people had conceived 5 years ago in this field. So definitely reaching some exponential growth here. So.
All right. So to wrap up, our final question is, how will synthetic biology make an impact on our lives? So to the average day-to-day person, how would you say that synthetic biology can make a significant difference? What is its effect?
So I think I would say that synthetic biology already is having an effect on people, in that even the products that we consume and the types of things that we're using are being built by processes that leverage synthetic biology technologies. For example, materials, clothing and foods that are, as well as drugs that are being produced, really require and leverage kind of advances in the synthetic biology field. But I think, at a very kind of a longer term timescale, I would say that we're really at a point in human history where we could begin to understand the compositional order of life and the structure and the design of how life is operates, and be able to kind of start to perturb that and change that and alter it in ways in which we've only been scratching at the surface of. If you think about kind of the major technological advances that have leapfrogged human civilization, things like agriculture, right? Being able to domesticate livestock these kind of big transitions. And I would say that synthetic biology is one of these is poised to be part of this transition where we're starting to be able to convert the natural environmental systems to enhance their properties in a variety of different ways. And so they're certainly going to be, very involved in important discussions and conversations about the, social impact of this type of endeavor. What are the benefits as well as risks that are associated with it? How do we kind of weigh those different axes and to be able to produce the kind of the most beneficial outcome for everyone, and how to democratize this type of technology such that it's not just benefiting a small section of the populace, but also something that could benefit humanity as a whole. So I think those are all conversations that synthetic biology the field is pushing towards engaging, and I think it's one of the more kind of open-faced, outreaching fields. I would say where really, because the end products are things that are going to essentially impact individuals, people in society, it really requires that open dialogue at the very beginning to think about what we should be making, what we should be producing and designing, and what we shouldn't be doing, and how we should govern and regulate, and to safeguard those kind of advances in a way that's responsible and just beneficial for all parties involved. So, yeah.
So thank you so much for joining us, Professor Wang. If anyone here listening to this podcast is more interested in learning about what Harris works on, they can visit his website, the site of his group. They recently published a really interesting paper about how recording events in cells with a CRISPR-Cas9 system. So you should definitely check out the press on that, or the paper. And so until the next podcast, this has been SBI with Professor Harris Wang. Thank you.